Hello, everyone. Today, we are going to learn about DNA replication, one of the most important biological processes in living organisms. DNA replication is the process by which a DNA molecule makes an exact copy of itself before a cell divides. This ensures that each new daughter cell receives the same genetic information as the parent cell. The main purpose of DNA replication is to maintain genetic continuity during cell division and to support the growth, repair, and reproduction of living organisms. Where and when does DNA replication occur? In eukaryotic cells, DNA replication takes place in the nucleus during the S phase of the cell cycle. In prokaryotic cells, DNA replication occurs in the cytoplasm. Steps of DNA replication. DNA replication occurs in several important steps. Step 1. Initiation. The process begins at a specific region of DNA called the origin of replication. An enzyme called helicase unwinds the DNA double helix by breaking the hydrogen bonds between complementary base pairs. As a result, two single DNA strands are formed, creating a structure called the replication fork. Step 2. Primer formation. Another enzyme called Primase synthesizes a short RNA primer on each DNA template strand. This primer provides a starting point for DNA synthesis. Step 3. DNA synthesis. The enzyme DNA polymerase adds new nucleotides to the growing DNA strand in the 5' to 3' direction. The new nucleotides pair with the template strand according to the base pairing rules. Adenine pairs with thymine, guanine pairs with cytosine. One strand, called the leading strand, is synthesized continuously. The other strand, called the lagging strand, is synthesized discontinuously in small pieces called Okazaki Fragments. Step 4. Primer removal and replacement. The RNA primers are removed and replaced with DNA nucleotides by specialized enzymes. Step 5. Joining the DNA fragments. Finally, the enzyme DNA ligase joins the Okazaki fragments together, forming one continuous DNA strand. Result of DNA replication. At the end of replication, two identical DNA molecules are produced. Each DNA molecule contains one original, parental strand, one newly synthesized strand. This is known as semi-conservative replication. Importance of DNA replication. DNA replication is essential because it ensures accurate transmission of genetic information, supports growth and development, replaces damaged or dead cells, enables reproduction, maintains chromosome stability, reduces genetic errors through proofreading by DNA polymerase. Conclusion. In summary, DNA replication is the process by which DNA copies itself before cell division. It involves enzymes such as helicase, primase, DNA polymerase, and DNA ligase. The process produces two identical DNA molecules, ensuring that every new cell receives a complete and accurate copy of the genetic information. Thank you for watching. If you found this lesson helpful, please like, share, and subscribe to Learning Center with Hanif for more biology and microbiology videos.